God bless you. And like to request you to wait patiently for this great talk from our colleague Daniel IGIRIMBABAZI. I'll be introducing him in a very short time before he speaks. In the seminar today, I am very happy to be in the seminar today with my colleague Daniel. And before we go on, I wanted to introduce him very deeply. Daniel is a PhD candidate in the Department of Soil and Environmental Sciences at the University of Wisconsin-Madison. He holds a BSc in Conservation Biology from the University of Rwanda and an MSc in Water Resource Management from the University of Wisconsin-Madison in the United States. He began his research career at the University of Rwanda Center of Excellence in Biodiversity and Natural Resource Management, where he contributed to biodiversity management as part of the Rwanda Biodiversity Information System, and he supported the specimen curation at the National Herbarium of Rwanda, and he helped organize the weekly seminars as I'm doing today. He later worked as a hydrologist for State of Wisconsin's Department of Agriculture, Trades, and Consumer Protection, contributing to statewide groundwater quality monitoring and assessment. From his master's training to the present, he has been actively involved in teaching and mentoring undergraduate students through instruction, training, and research support across biology and soil science contexts. His research focuses on winter climate variability and its effect on forest, soil structure, and microbial process, with emphasis on freeze-thaw dynamics and soil aggregate stability in temperate forest system. Daniel aspires to understanding of seasonal soil health dynamics and Daniel also aspires to understand seasonal soil health dynamics during the often understudied winter period in cold region ecosystems. Without further ado, I'd like to welcome my colleague Daniel. So he takes us through the presentation under the title Winter Dynamics of Below Ground Biophysical Processes in the Two Temperate Forest Soils in Wisconsin, USA. You are welcome, Daniel. I'm very happy to welcome you as a colleague and then an expert in this area. And I'd like to just mention that I'm happy with the level you are today. Thank you very much. We would like to hear from you.
All right. Thank you so much, Vena, for the sweet introduction. And welcome, everyone. Today, I'm going to be talking about winter dynamics of below ground biophysical processes in two temperate forest soils in Wisconsin, USA. So, just a disclaimer, if you can't hear me or something, please feel free to let me know or write in the chat. Want to make sure we're on the same page. All right, let's get into it. Before I start, briefly, I also want to express my appreciation to the center. I always felt like I was never going to be in this situation to be the one presenting, and here I am. So I'm really excited, and Thanks a lot for a great job you're doing at the center. So I'm gonna be talking about winter time, and some of you might be like, why is this guy studying about winter and snow when he's from Rwanda? So I thought I might put these together for all of us. So basically, on the left side, you see Karisimbi. Growing up, I grew up in a um, village nearby Karisimbi, so this was my usual sight in the morning. I always felt curious to know if what that white stuff was, but I didn't have the opportunity to get there because it was logistically difficult or almost impossible to get there and know what's going on at the top. So when I was in the US, I kept my curiosity from childhood because I always wanted to interact with snow. And when I was browsing online, at one point, I got this photo at the top online at a certain blog by two brothers, Eric and Matthew. They're from the US. And I emailed them and I said, it looks like you guys been to Mount Karisimbi. Is that really the actual snow that we know? And they confirmed that they've been there. They are the ones who took this photo. And they say, yes, this is snow at Mount Karisimbi. So this is always a really nice segue for me to enter into the research I do about snow because it's an aspect I've always been curious about and trying to understand how snow can interact with real life aspects like soil and what we eat is really fascinating to me. So I'm happy to share that with you guys. All right. So this is the outline of what we're going to talk about today. Quickly, it's going to be about background, mostly talking about what we know about snow and what it means in terms of its interaction with the soil. And we will talk about the gaps in the literature, and we will talk about the research questions and hypotheses that I'm trying to explore. Experimental design, we have preliminary results because this is a long-term study. I only have results for season 1 out of 3. So that's what I'm going to be sharing today. And we will go ahead and do Q&A. All right. Now, these are the key points I need us to understand at the get-go. This is ecological significance of seasonal snow. So when I talk about seasonal snow, this is because the region I live in, it's called temperate region. So that's in the northern part of the United States and some parts of Europe. But today I'll be focusing on Wisconsin, which is part of that region. So we have 4 seasons. We have fall, winter, spring, and we have summer. So whenever you see seasonal snow, think of it as snow that falls within the cold seasons, which means winter and spring. So that snow acts as thermal insulation. So basically, it acts as a blanket to the ground, to the soil. This is counterintuitive because initially I thought if you see snow on the ground, that means the ground is cold. But actually, that's not the case. So whenever you see snow on the ground, think of it as it's protecting, it's making the soil warm. So that's important for us to know at the get-go. So that protection of the ground leads to healthier soil structure or structure stability. This is because it reduces cycles that we call freeze-thaw cycles, and I'll talk about them later in a minute. Basically, those cycles can be damaging forces to the soil. So when you have the snow on the ground, those cycles reduce and the soil becomes healthier. Speaking of snow, again, these are projections. of future climate in Wisconsin. This is a map of Wisconsin. We're just gonna focus on winter and spring. So here at the top left, we can see that by 2050, winters are gonna be warmer and spring during daytime, and even warmer in nighttime. So point here is it's projected that Wisconsin is gonna get warmer in cold season. And here on the right side, we have precipitation, so the rain. So by 2050, it's expected that Wisconsin, again, let's focus on winter and spring, our cold season, it's gonna get more precipitation. So what this means is this precipitation will become warmer in winter and it won't form snow. So we will have reduced snow because whatever water will be formed, it will melt right away. So what happens is, look at this diagram on the left. Here you have water in blue. This is soil aggregate. So when it's cold season and you have water falling on the ground, it freezes because it's winter time. And when it freezes, that water expands as an ice. And when it expands within soil, it breaks the soil structure apart. So this is reducing aggregate stability. So you need a healthier, stronger soil that's not cracked like this. So this phenomenon is what we call freeze-thaw cycle. So this is simply showing that when water gets in the soil, it freezes and then it thaws. Thawing is like unfreezing, becoming warmer. So when that repeats over time, the ground or the soil structure gets broken apart. Now, with that information in mind, please keep the freeze-thaw cycle in mind related to soil, because we're going to talk more about this moving forward. So this is a study that was published in 1995. So they tracked the snow cover in the Northern Hemisphere from early 70s to early 90s. On the right, what I need you to focus on is you can see that from early 70s to mid 80s, we had on average snow cover was above normal. So that means the ground was protected. But starting from late 80s to early 90s, you can see that mostly snow cover is below average. So that means we are having more of these situations, freezing and thawing, because the ground is not protected from the fluctuating air temperatures in the cold season. So the point here is since 70s, there was snow variability, but mostly reduction in the snow cover as the year passed. Nowadays, these are just publications, not official. These are web page publications that talk about warming winters. So the idea here is winters are becoming warmer than usual. That means snow formation is going to be reduced. So we're going to have less snow cover on the ground, and the ground is going to be losing that protective insulating cover by the snow. And this study that was published in 2024 shows mostly what happens when you have variability in the snow cover. So basically, what it says is the climate change leading to global warming, and during winter season as well, that season is affected in a sense that when you see a downward arrow, that means that aspect is reducing. When you see an upward arrow, that means that aspect is increasing. So you might not be seeing this clearly, but we have snow here at the top. It's it's in the gray color. So the warming climate in winter is leading to this snow extent to decrease, and its albedo capabilities, or the ability for the snow to reflect the sun radiation, will decrease, and the depth of the snow will decrease. Of course, it will melt, and its duration on the ground will decrease. And what that will mean is, in the ground, this is the ground, I will have more increase in temperature fluctuations because the ground lost its insulating ability in the snow. And we will have more liquid water availability in the ground. That might be a bad thing, as we will see later. We will have an increase in the freeze-thaw cycles. Remember these ones? They break down the soil aggregate stability. So they will increase with increasing climate change or warming winters. So what's in the green is related mostly to biological aspects or effects, but we're not going to go into this. But this is also part of my research. For today, I focused on the physical aspects that will happen in the ground. So takeaway message to this slide is snow variability influences freeze-thaw cycles, which leads to cascading effects on the soil structure. Now, diving into different studies that quantified what I'm talking about. This is a study that looked at the soil series. You can see on the x-axis. It's a busy diagram, but I'm gonna walk you through it. So let's just focus on the last two bars per each series. So on the y-axis, we have aggregate stability. For this series, the black bar here and the white bar. So the black bar is the stability of the soil before freezing and thaw cycles. You can see it was higher, but then after this soil has experienced freeze-thaw cycles, this is the end result. So you have reduced aggregate stability, and this process was the same in all these series under study. So here, what they found is when you have snow removal or reduced snow, you will have more freeze-thaw cycles, and that will overall lead to reduced aggregate stability. Now, some other studies wanted to know, okay, if that aggregate stability is reduced, what comes next? What happens? Does it remain low stability, or does it bounce back? So what they found is, like this study by Ed Boswell, who is part of, he did his PhD in my research group, so we know each other pretty well. So he found that aggregate stability decreased, but at the end of summer, they return to their original set point. So think of it as here at the top. If this decreased aggregate returns back to the normal, and then and so on. But he did this in agriculture and prairie systems. Another study say that this aggregate stability decline after freeze-thaw cycles can actually lead to a new equilibrium in cell structure. So that means if the aggregate might remain low stability without bouncing back. So this is an interesting aspect or an effect of freeze-thaw cycles, but generally more freeze-thaw cycles reduce aggregate stability, although some other studies may find increased aggregate stability, but mostly it's reduced aggregate stability. Now, with all that information in mind, I wanted to highlight that most freeze-thaw cycles are studied or conducted in lab simulations, because these studies might be hard to manipulate or to work with in situ or on the field. So basically, the results they find is mostly because they were able to control all the aspects they wanted to. So basically, in lab, you can control how much temperature you put to your soil, how much moisture, how much, um, let's say, freezing aspect. So that might not necessarily reflect what happens on the field. That's one research gap. Another research gap is fewer freeze-thaw cycle studies are conducted in temperate region that I will talk more about later. So to our knowledge, there is no in situ studies that examine the effect of freeze-thaw cycles on temperate forest soils. So that's the gap my whole research is trying to address. So I talked about temperate region. So I want to dive back into it a little bit. So on this map, you can see above this dark line and below it, this region is 60%, and it's of land of Earth, 6% of Earth land, and it's seasonally covered by snow. So you have snow on this region in winter and spring. And what's most important here is that this region again below this line and above this line, and that's where US is included, and Canada and most part of Europe. So it holds 70 to 80% of global terrestrial carbon. So that's really important region in terms of carbon sequestration. And to top this, if I overlay this map to the same region, you can see most of it is covered by forests. So it has a high significance in terms of terrestrial carbon sequestration. So this is important, and why I wanted to dive into the effect of snow variability to this region. Now, because this is a huge region, I was not able to study all of it. So I focused on just part of it, which is Wisconsin. And a state of Wisconsin is also facing changing climate and, of course, variability in snow cover. So some parts of the state are becoming warmer in winter and having low snow cover. That means more freeze-thaw cycles, and that has some effect on the soil, as we have seen. So I wanted to dive into this state. As you can see on the right, it's showing the maps, the dominant soils of Wisconsin. You can see most of it is red, which is a soil order called alpha-soil, mostly clay, and some part of it is dominated by, you can see blue, it's the spodosols. So those two soils are the ones my research focused on in Wisconsin. This is just a general overview of where those soils are located or distributed in the US. So again, my research will focus on alpha soils and spodosols in Wisconsin. So my research question is, how does soil aggregate stability measured in mean weight, diameter, and wet aggregate stability, we will talk about this later, responds to freeze-thaw cycles in two forest soils that we have seen with typical and not typical vegetation. So basically, if you have alpha soil that has its typical forest type versus if you have alpha soil with non-typical forest type, how will those two systems respond to freeze-thaw cycles when the snow is variable? This is a hypothesis. We hypothesize that continuous snow cover will increase the aggregate stability compared to where you have less snow or continuous snow removal. This is the hypothesis. So I'm going to dive into the area or study area so that you all know. I briefly talked about Wisconsin. That's the location. I understand maybe a lot of you do not know necessarily where it's located. It's in the region called Upper Midwest of the United States, close to Minnesota. We have Illinois and Michigan. I'm gonna make this easier for you all. I'm pretty sure a lot of you know Chicago. So Wisconsin is pretty close to the city of Chicago, just for context. Now, in Wisconsin, we have various counties. My research focused on two of the counties, one in the northern part of Wisconsin called Oneida County. That's where we have the spotted soils, the sandy soils. And I have another study site in the southern part of Wisconsin, Dane County. That's where we have the alpha soils, the clay soils. All right, so let's dive into one at a time. So starting with Oneida County in the northern Wisconsin. Um, this is the spotted soil dominating that area, that county. This is pretty much how spotted soil looks like. It's mostly found in cool, moist regions. Um, it has a sandy structure and has organic matter accumulation in the B horizon. This soil, this spotted soil, can also have moderate to deep organic horizon. It has, because the common vegetation or the forest type in this region is coniferous forest. Think of this as pines that have needles, needle leaves, as you can see on the ground. These needle leaves are hard to decompose, so they accumulate over time. All right, so keep that in mind. That's the spotted soil, and it's 17% of Wisconsin land area. So on this soil type, we have two sites. We have coniferous forest on spotted soil, and we have deciduous forest on spotted soil. I color coded these in green and red to show you that here in green is ideal because you have ideal vegetation or forest type on ideal soil type. So because spotted soils ideally have coniferous vegetation or forest, but part of this county also has some deciduous trees growing up on it, and we were curious to see if that will have an effect on its response to freeze-thaw cycles compared to this ideal situation. So here we have same soil type, spotted soil, just different forest types. Let's keep that in mind. And in the south, at Dane County, pretty much have the same setup, but different soil type. We have alpha soil, which is 47% of Wisconsin land area. And this is pretty much basic characteristics of the alpha soils. Just the main important thing is it's clay soil, and common with or together with deciduous forest. So it's the opposite for the spotted soil. So in this area, we also have two sites on alpha soil, so non-ideal in red and ideal in red. So what we did so in total, I have 4 research areas throughout Wisconsin on 2 soil types. So at each of the locations, this is the experimental design we conduct during winter. We insulate the soil, or we warm the soil to mimic a future where you have more snow on the ground. And that means we'll have more blanket or more protection of the ground. And then we have another treatment where we remove the snow continuously with a structure that limits snowfall on the plot. So this is a future where you have warmer winter with less snow on the ground. And we have control or ambient. This is simply to show the type of material we use here. It's called straw or hay. We put it before winter time, and throughout winter we use it to warm the soil. These are the undergrad assistants in my research lab, and this is how we make sure no snow is falling on the ground. So we manually remove the snow when it accumulates on the roof, just to keep it away from falling on the ground. All right, so insulated, no snow, and control. So in each of these plots, we do a lot of things. We measure covariates of soil aggregation or things that may impact aggregation. So moisture, temperature, frost depth, snow depth, and we have various instruments for each one. This one is, we call this TMS, measures temperature. This one is for moisture. It's called CS616 by Campbell Scientific. And we measure soil temperature using what we call type T thermocouples, just some wires at different depths in the ground to track down temperature changes from our manipulation. We have frost tubes that measure how deep the frost is in the ground. And we have snow depth to measure how deep the snow is. This is snow gauge. And all this information is conducted or transferred to a central location that we call CR1000, where I hook my computer and see all the data or download them. This is one of the examples of the data about temperature in our treatments. Pretty much this dotted line in the middle is 0 degrees Celsius. So where we insulate the soil in winter, you can see that this ground or the soil underneath remains above 0, exactly how we want. This is control, it goes up and down. And for the no snow, because you want the soil to have no insulation, to be colder, you can see that it's mostly below 0 as we want. So this is an example of all those instruments and what they help us understand. All right, for methods, we have completely randomized design. I talked about our treatments. We have 3 replicates of each one. We use two-way ANOVA for now because we're still early in the process. With more data, we will have different models to look at the results. And so aggregation is measured using method by Yoder and Kemper and Rosano. I will explain about this in a minute. So what we do is we collect soil samples in those plots before and after winter. So basically in the fall and at the end of spring. And we collect soil samples, 2 cores in each plot. We composite them in a bucket and we transport them to the lab where we conduct soil aggregate stability using the methods I described earlier. This one is different sieves sizes that helps us understand the mean weight diameter. And this is what we call wet sieving apparatus, helps us to understand wet aggregate stability, or the ability of the soil to withstand rainfall erosion, I would say, for example. Now we have preliminary results. This is just for season 1 out of 3 seasons, winter seasons that we'll have to do. We are done with actually season 2, but we haven't finished all the data interpretation yet. And we have one more season to undergo. And then we'll combine all the results and answer our hypothesis. So for now, these are simply what we are seeing so far. Starting from northern Wisconsin, Oneida County, remember the place with the sandy soil? I focused on just the ideal situation. So conifers on spotosols. So what we found is for our two aggregate stability proxies, let's start with this one in blue, the wet aggregate stability. This is pre-treatment, and these are our treatments on the x-axis, ambient, insulated, and no snow. So this is pretty much how stable the soil was before our treatments. And this is how it changed after one season. So from, let's say, November to April. So mostly you can see aggregate stability increased. Of course, we have more increase in the insulated plots because we have less freeze-thaw cycles in it. And what's interesting here is that after winter, so basically after spring, throughout summertime, this is what happens. So aggregate stability decrease throughout. This is pretty much the same results when it comes to this proxy, the mean wet diameter in green. So you have an increase in aggregate stability, of course, more increase, a little bit more in the insulated. Then you have a general decrease during summer. And we will talk about what that means. But again, this is preliminary. We will have more than one season. On average, we might have a different result. All right, for the southern part, Dane County, again, on ideal forest type, deciduous on alpha soil, we have pretty much the same trend that we've just seen. So this is pre-treatment for the wet aggregate stability. So during winter, you have a general increase in aggregate stability, and in summer, you have a decrease. Same with the mean wet diameter. So you might be wondering what's driving all those dynamics. First of all, this is suggesting the buffering capacity of forests. Forests have a high buffer capacity, so they take time to respond to treatments. So this being just season one, the results are not necessarily confirming our hypothesis, but it's still too early. And what we're seeing in terms of decrease after cold season, decrease in aggregate stability, some studies found pretty much the same, and it's linked to what we call wet and drying cycles. So that means when summertime includes some sort of precipitation, especially in that region, the more precipitation or more moisture has been linked to less aggregate stability by different studies. So that summer, where you see a lot of decrease, showed some more precipitation patterns that influenced decrease in aggregates, because this motivates some microbial activity, and they break down different aggregates in different ways, because decomposition can lead to less aggregate stability, although in some other aspect, it can increase aggregate stability. That's a lot of microbial part that we're not going to go into. But I just wanted to point out that that has to do with decomposition in summertime, leading to less aggregate stability. All right. Now, just summary. This is, I talked about a lot of things, but these are key takeaways that I need us to understand. So climate models predict that in the future, temperate region is going to get warmer in winter time. So that means um, the snow cover and snow duration in winter is going to be really decreased or reduced. So what that means is the ground that was usually protected by that snow will remain um, without protection or without insulation. And that will lead to more freeze thaw cycles, those physical processes that lead to aggregate stability decline by reducing cell structure. All right. And that will have important aspect in the temperate region, especially that they are understudied. So that's why understanding the impact of this changing winter climate to soil health in this region and in situ is really crucial and it's important. And that's exactly what my research is doing. So with that, I want to thank my committee members. I have a lot of committee members, as you can see. My funding agency, United States Department of Agriculture, or USDA, and the research stations where my plots are located at, and different people who help in different aspects during my research, and of course my department. And here at the top right corner, Bolster Lab, that's my research group. Really super dynamic and helpful group that I'm really happy about. And with that, thanks everyone, and I will take the questions.
Yeah, thank you very much, Daniel. That's very nice. And if I can summarize, I can see that your research is speaking more about the impact of climate change on soil stability, especially in the temperate region, where the soil is seasonally covered by snow.
Exactly.
Yeah. Then my colleague in the chat room is saying, can you explain clearly the difference there with African continent?
Yes, that's a great question. Um, so I'm trying to look for the chat to make sure I address the question correctly. But straight up, the difference is think of less snow variability or a lot of snow variability, basically warmer winters as a disturbance to the soil. Because when it's winter time and you have snow on the ground, that snow is protecting the ground. But in Africa, we do not have snow, although, as I showed, we only have snow in a few areas, so like Mount Kalisimbi and I heart, some country in southern Africa
Yes.
that has some snow. But mostly in Africa, in tropical region, uh, you need to replace snow with rain variability, so rainfall. So some seasons you have a lot of rain, some other seasons you have less rain. So what that means is when you have a lot of rain, that can have cascading effects on the soil. It may lead to soil erosion when your soil structure is not stable. Uh, so basically, in Africa, there is no disturbance necessarily related to snow, but there is disturbance related to rain. Think of river Nyabarongo. It takes soil all the time. It's always dark because it's grabbing all the soil organic matter from the nearby fields. So that's a disturbance. And we need to understand how the soil nearby that river Nyabarongo um, can withstand that that water. So when you look at the mean wear diameter and weight aggregate stability in that soil, you can understand that over time, that soil will eventually, all of it will be damaged, or maybe it will not necessarily be too damaged. So that's the so the only difference is um, in Wisconsin or in the US, you have snow because it's cold seasonally. But here, you have more precipitation hitting the ground directly, which is also a disturbance to the aggregate structure. Did that answer the question?
Let's hope. Yeah, he says very good. Keep blessed. It's well understood. Yeah, he understood
Awesome.
it. Yeah, it's very interesting, and I'd like you to do the same research at the Mount Karisimbi, Mount Kilimanjaro, and South Africa to see
Yeah.
the difference maybe in the future.
Yeah, it's something I'm really curious to explore further as well. So, yeah, definitely cool research ahead.
Yeah, actually, this research shows that climate change is far-reaching and it has a lot of impacts. And I am thinking that some of them are not even explored.
Mhm.
And I'm wondering if at the government level, I'm speaking about the government of the Wisconsin state, do they do something to reverse the climate change, at least at the state level?
So yeah, that's a good question. Basically, as we know, it's hard to reverse climate change. What they do is to mitigate climate change, and mostly they start with investing in research, exactly what I'm doing, to understand what's happening to the environmental aspects when you have climate change. Because another part I didn't mention because of time, the northern part of Wisconsin is um, nowadays has more snow than the southern part of Wisconsin. Um, Although a lot of agriculture happens in the southern part of Wisconsin, Wisconsin is considered America's dairy land, so they are really heavy on agriculture and uh, um, raising animals, cows. So they are really interested in understanding how climate change will affect all those parameters. So what they do is invest in research and trying to find ways to mitigate climate change by modifying agriculture, doing some smart agriculture um, aspects, planting crops that can tolerate the changing climate. It's already being implemented. So I would say Wisconsin is up front when it comes to uh, climate adaptation and climate change mitigation.
Oh, wow. It's very nice. Yeah, I want to receive one question or comment before I close the seminar today. Kindly, yeah, Nsenganeza, you are welcome to speak.
Thank you very much, Vena. Thanks to the presenter for good presentation. Very interesting. Are you getting me?
Yes, very well.
Okay, thanks a lot. I would like to ask one question, Daniel. Daniel,
Mhm.
did you also consider also sampling soil nutrients along soil horizons? Uh, if you were interested in your research also to measure those nutrients, could be a good meaning for the science. And also, if for instance, if when you are going to collect soil samples in where there is snow, did you take like a time to remove all snows, and if possible, could you tell us, for instance, the cover of the snow, the height of the cover of the snow before you reach the soil sample? Thank you.
All right, good questions. So I'm gonna start with your first question about sampling for soil nutrients. So the answer is not necessarily, not actually the actual nutrients, but I measure the nutrient cycling indirect. I have what we call um, litter decomposition project, where I track down how different litters in those forests on the ground how they decompose over time by what microbes. But also to answer your question in a better way, what I presented is part of my first chapter. Second chapter is heavily focused on microbial response to all those changes I mentioned. So we do what we call microbial population. We track down in our soil treatments how much microbial activity is happening. So during winter, I continuously measure soil respiration to see how um, fungi and bacteria are responding to the fluctuation in temperatures. So I use some instruments called soil chambers that you put on the ground, and you see the CO2 being released. And then I put some samples in the lab to measure for what we call PL, phospholipid fatty acids, to understand if you have maybe more actinobacteria, or if you have gram positive, gram negative bacteria, to use all that information to link to the aggregate dynamics, because um, it has been proven that all those changes have microbes have a big part in the changes, not just the freeze thaw cycles. So to answer your question, I do not measure nutrients directly, but I do measure microbial behavior that can answer part of the nutrient dynamics. For your second question, I do not take samples during winter. I take samples before and after winter. So before winter, to understand how the soil structure is looking like. So around November, we have advanced um, weather prediction models that can tell me, okay, winter time is going to start next month. Now I need to collect samples. And then I analyze those samples for before winter. And then after snow period, so around end of April, when there is no longer snow on the ground, I go back, collect some samples for the after winter, and then I compare before and after results. Um, so yes, I do not collect samples during winter, but I track down the snow depth to answer part of your last question. I showed a photo with a snow gauge. It's pretty much like a big ruler that we put in the ground. And throughout the season, we know, okay, maybe in November we had less snow. In December, we had this snow. In January, we had less snow. Then we can compare all of that to the results we are seeing with freeze thaw cycles. But I would say in northern part of Wisconsin, like this past winter, the snow was pretty much on average like 12 inches or 1 foot of snow. Uh, you're gonna, I apologize because I don't know that in centimeters or whatever. Um, yeah, so 1 foot of snow on average in northern part. In southern part, Dent County, very minimal snow on average. So like 3 inches of the snow. And yeah, did that answer your question?
Yeah, thank you very much, Daniel, and thank you, colleagues, for attending the seminar today. Allow me to close the seminar by here, and before closing, I'd like to quickly thank Daniel IGIRIMBABAZI for speaking to us and for this great presentation. Yeah, he is a young man who is committed and he grew very quickly because I know Daniel from the third year of his BSc studies at the University of Rwanda. I know him when he came to do the professional internship at the center and later on working for the Arabs. Yeah, actually, he's a good man to learn from, and young man here can also connect with him so he can share some opportunities if available. Yeah, speaking to us is an opportunity to learn science and also to learn from you professionally.
Yeah, I really appreciate it much,
Yeah, thank you very
and I
much.
always
Yeah,
feel for our set.
yeah, and if
Yeah.
you are okay with it, you can drop your WhatsApp number in the chat room. I know some people here may need to talk to you, ask you questions. Yeah, connect. Yeah, if that's okay
All
for you.
right, and yeah, to be to make this a safer aspect because this recording might be put online. So I'm gonna
Mm-hmm.
say LinkedIn information is better. So pretty much my full name, Daniel IGIRIMBABAZI, in LinkedIn. You can add me, I will add you back, and we will talk. So I'm
Okay.
gonna write my name here.
Yes,
Yep.
yes.
Yes.
Great, great.
And yeah, thank you everybody for coming and looking forward to learning more about you as well when you present.
Yeah, thank you very much. Yeah, to my colleagues, thank you very much for joining. Let's meet again next time when we have a seminar like this.
All right. Bye.
Keep in touch. Bye.